Hello and welcome everyone. Thank you guys so much for tuning in to our next edition of Café Sci this evening. My name is Colin Summerhauser and I'm the head of Haunted House in Palo Alto. Now I know most of you know this already, but just in case we have some new faces tuning in tonight, Haunted House is a co-working space and cafe, and we also offer free science and technology events such as Café Sci to the community on a monthly basis. So over the course of the last month or so, we have taken Café Sci virtual for the first time in its history due to COVID-19. And it's been very exciting because we have been able to engage more people than ever before thanks to the for our presentation this evening, remember to use the interactive question widget on the bottom of the screen to ask questions throughout the presentation, and be sure to upvote the questions you would like to see answered. And last quick reminder, all of you will receive a post-event survey following the event. Please, if you could, just take 1 minute to fill it out. It'll be a tremendous help so we can understand how we can continue to improve these sessions as we move into the future. And now with that, I'll hand it over to our Café Sci lead, Kay, to introduce our amazing speaker today.
Hello, welcome to you all from Café Scientifique, Silicon Valley. Next month, on third Thursday of July 16th, we look forward to hearing from Dr. Michael Fischbach of Stanford on the interesting topic of microbiome. Hope all of you can join us. But today, we are going to hear about epigenetic inheritance from Professor Susan Strome. She is the distinguished professor in the Department of Molecular Cell and Developmental Biology in University of California at Santa Cruz, or UCSC. Dr. Strome received her BA in chemistry from University of New Mexico in Albuquerque, and her Ph.D. in biochemistry from University of Washington in Seattle, and then served as a postdoctoral fellow at University of Colorado in Boulder. Susan's teaching career started as a professor in the Department of Biology in Indiana University in Bloomington. In 2007, she joined UCSC. She now serves as the distinguished professor and the chair of the Department of Molecular Cell and Developmental Biology. The focus of her research has been on epigenetics of inheritance. Using model organisms, her lab has established new paradigms for establishing and maintaining germ cells. Dr. Strome has been awarded numerous fellowships, awards, grants, including Guggenheim Fellowship, Wallace Franklin Medal, and UCSC Presidential Chair Award. But luckily for us, she has received many awards for teaching excellence. The first one was from the Department of Biology in Indiana University, then from the trustees of Indiana University, and recently from UCSC. Dr. Strome has developed innovative methods to teach genetics to undergraduate students, using common objects such as pipe cleaners or pool noodles to represent chromosomes. The students can enact some of the genetic processes, increasing comprehension through participation. I'm sure her classes are engaging, inspiring, as well as fun. Wish I was a young student again. Here is Professor Strome.
Great. Can you see me? I'm live? Good. I'm really happy to be here. I was hoping that I would be here in person, joining you at the Haunted House, but COVID-19 is keeping most of these meetings virtual, and we're learning how to run with it. So I'm excited to bring you the story virtually, and I've lined up a few dolls in front of me to act like I'm talking to people. So let me share my screen. Let's see. How's that? Can you see my screen? Great. I'm going to talk to you today about epigenetic inheritance, and I'm focusing on the way that we send memory information across generations. Here I'm showing you parents, the young me, the young my husband, and our offspring received information from us. not only genetic information, but epigenetic information. Now, I try to dissect epigenetic mechanisms using a model system. And in my case, that is Caenorhabditis elegans that I'll introduce you to. So let's start. The roadmap for this session will be, I'd like to present a bit of a primer on the genetic material that's transmitted by germ cells so that we're all on the same page, and then tackle the question, what is epigenetics? Does epigenetic regulation influence human health? Why do we study epigenetics in model organisms instead of humans? And then I'll tell you some of the approaches and lessons that we've learned from our model organism. So I drew out this cartoon of the chromosomes, the set of chromosomes in one of your cells, to give you a sense of the genetic endowment that is present in each of your cells. And this would be one of your non-reproductive cells. This could be a muscle cell, a nerve cell, an intestinal cell, a skin cell. In each cell, you have 2 versions of chromosome 1, 2 versions of chromosome 2, 2 versions of chromosome 3, etc. The chromosomes are not color-coded, by the way, but it helps me keep track of them. Each chromosome has about 1,000 genes on it. And so these 2 versions of chromosome 1 have the same genes, but certain copies of the genes come from mom and certain copies of the genes come from dad. And then we have these 2 X chromosomes that a female has and an X and a Y chromosome in males. Now, you inherited 1 of each chromosome from your mom and 1 of each chromosome from your dad. And the inheritance was via germ cells. In other words, eggs and sperm. So here's a scanning electron micrograph of an oocyte. That's this is a human egg, a human oocyte, and a lot of human sperm surrounding it. By the way, they're not pink. They were just color-coded for ease of viewing. A human egg will be fertilized by 1 sperm to generate a baby. And the rest of the sperm are blocked from entering the oocyte or egg. So it's via this big oocyte or egg and these tiny sperm that chromosomes are sent into the embryo. So here we are, how to make babies. We have the union of an egg with 1 of each chromosome and a sperm with 1 of each chromosome. And after they unite, we're back to that starting cartoon that I showed you, where each cell has 2 copies of every chromosome, 1 from mom, 1 from dad. So we get all this genetic diversity, and furthermore, we get inheritance from both of our parents. So what is epigenetics? I imagine the term is somewhat familiar to you. I'd like to develop a working definition for this evening and then address whether there's evidence that epigenetics is operational in humans and how we study it in model systems, and in our case, worms, and then what lessons have we learned. So what is epigenetics? My guess is that many of you have heard of it. It stands for on top of genetics. One of my favorite definitions is from Denise Barlow, all the weird and wonderful things that cannot be explained by genetics, which is fun and playful, but is actually not very helpful. I would instead like to develop it this way. We think of genetics as regulation by the DNA code, regulation of which genes are expressed, regulation of your appearance and your behavior and your health by the DNA code. And by that, I mean, here's the DNA shown in purple, the DNA double helix. We'll get into this wrapping a little bit later. The DNA sequence, the DNA code is here, GC base pair, AT base pair, TA base pair. This is the DNA, the genetic material that you inherit. It's super important. Epigenetics is regulation on top of that. So it's regulation of which genes are expressed, how you, what you look like, your health, your phenotype, by how the DNA is packaged and how the DNA is deployed. So let's consider the packaging of DNA. DNA, as its first level of packaging, is wrapped around these beads called nucleosomes. These are made up of histones. DNA can be further wrapped and compacted, as shown here, by forming coils and loops to enable it to fit inside the nucleus. And this packaging is really, it serves two really important roles. One important role is compaction. The DNA in each cell of your body is about 6 feet long. So 6 feet of DNA has to fit into that tiny little nucleus in your cell. This packaging enables that compaction to happen. And to give you a sense of proportion, this actually looks like a basketball, doesn't it? This nucleus. Imagine that a nucleus was the size of a basketball. The DNA contained in it would extend from about Hana House to Santa Cruz. Okay, so there's a big packaging need. Along with compaction of DNA, it has to be regulated so that genes that need to be transcribed into RNA are open and can be accessed and transcribed. And genes that might reside in this more compact chromatin fiber are more tucked away and will be kept what we call repressed or silent. Now, what are the major epigenetic mediators? The most commonly studied epigenetic mediators are direct methylation of DNA. So Cs, cytosines, can be methylated. And regions of the genome that have cytosine methylation are regulated differently than regions of the genome that lack cytosine methylation. So that's one epigenetic mediator. Another is histone modifications. I told you that histones make up the nucleosomes here, these beads that DNA wraps around as its first level of packaging. These little tails on the histones are subjected to a whole variety of covalent modifications that impact how the DNA is packaged and how the DNA is expressed. And we'll get to the whole point of gene expression in a few minutes.
What's wrong with you?
And then there are small RNAs. And small RNAs can float through the nucleus here and can impact which genes are transcribed into RNA, in other words, expressed, and which genes are kept silent. Okay?
Take the box out, so that might make it make difference.
So there are two types of evidence that epigenetics influences humans, and I'll tell you sort of the within generation evidence and then across generation evidence. One type of evidence comes from studies of twins. All right? So identical twins have identical DNA sequence. And you would expect them to look and behave and develop identically. And indeed, when identical twins with identical DNA are examined when they're young, they tend to look and behave very, very similarly. But they may age very differently. They may experience disease, stresses in the environment, and their outcomes later in life may be quite different from each other. Fraga, in 2005 and in other papers, found that, in fact, the packaging in young twins was very similar, while the packaging of DNA in older twins was beginning to diverge. And that illustrates that their life experiences can impact DNA packaging and can have influences on how they look, how they behave, and on their health. Now, can epigenetic information be passed from parents to offspring? This is really the topic of my session tonight. And the model in humans would look like this. As children age and grow up into moms and dads, their life experiences and environment can impact, it can affect the DNA that is packaged into the egg and the DNA that is packaged into the sperm. So that when those two germ cells unite to give rise to a baby, an offspring, that different packaging can influence the health and longevity of the embryo. Is there evidence that this model happens in humans? Does this happen in humans? Indeed, human studies suggest that lifestyle and diet can influence the health and longevity of descendants. This was a fascinating study. It was done mainly by Pembrey, Marcus Pembrey, in Överkalix, a northern province of Sweden, where they have detailed records of food availability and the health over generations. And so the analysis that Pembry did was to study the effects of food availability in the early 1900s on the health and mortality risk of the individual's offspring and grand offspring, okay? And he observed a link between boys' food supply back in the early 1900s and their grandsons' mortality risk, and a link between girls' food supply in the early 1900s and their granddaughters' mortality risk. Really fascinating, right? Across 2 generations, and there's this sex bias. But this is simply a correlation. It's a link. And we certainly cannot analyze cause-effect in humans, and we cannot analyze mechanism. We can't try to dissect the mechanism that underlies this. And by the way, there's a wonderful YouTube video, a wonderful PBS Nova program called Ghost in Your Genes from back in 2006. So jot that down if that sounds interesting to you. It features Pembry, and it is all about epigenetics, and in fact focuses on this over calyx and other studies. It was fun to me that when my mother watched the PBS special on Nova back in 2006, she got all excited and called me and said, "Susan, have you ever heard of epigenetics?" I said, "Mom, that's what I study." But evidently, I hadn't made that clear enough to her. So after that, she thought what I study is really cool. Okay. So the question becomes, why study epigenetics in model organisms? I've said that there's some suggestion that it happens in humans. Why not study it in humans? Why study it in model organisms? And by model organisms, I mean the premier, the most heavily studied model organisms are the nematode, see elegans, the fruit fly, Drosophila melanogaster, mice, rats, zebrafish. So I'm putting this question to you, okay? You, the audience. Why study epigenetics in model organisms? Now, there's some delay between my saying this to you and you're being able to enter your answer in the question toolbox. So talk to your partner. We do, in my classes, we do a lot of what we call think, pair, share. Think about my question, talk with your partner, and then share your answer. And so in the question tool, I'd like you to submit some possible reasons why we would study epigenetics in model organisms. And while I wait for that delay for you to enter some answers, I will just show you what our model system looks like. So here's the tiny nematode worm, see elegans. Okay, it's swimming across the screen. I'll let it swim once, and then I'll go back and show you a few things. Okay, let's let it swim, and then I'm going to stop it. So this worm looks larger than life on your screen, but it's actually 1 millimeter long. It fits on the head of a pin. One thing you can see is it's transparent. We can see through it. The region here that looks sort of granular is its intestine, okay? Its germline is back here and is not very distinctive. What you'll notice here are a bunch of football-shaped objects, and those are the embryos. Those are the offspring of the worm, and right now they're still living in the worm, but they will pop out this opening right here, and then they'll continue their life outside the worm. Okay? So now I want to return and see if I can access your answers to why study epigenetics in model organisms. So I'm going to try this. I'm looking. Okay, great. Someone said, because it's a whole bunch faster. Spot on. Worms, so think about the time it takes for an animal to make babies. So from the time we are born, we're a baby, until we grow up and we make babies is anywhere from 14 to 18 years until we can make babies. And it might be a lot longer depending on when we want to start making babies. In worms, it's 3 to 5 days. Okay, from the time a worm baby is born until it grows up and makes its own baby. So it's fast. Legal issues with people. Excellent. It is not permitted, it's unethical to do experiments on humans. We don't have such ethical issues when we do experiments on worms, fruit flies. We do have ethical issues when we work on mice, and we have to make sure that we are treating them in a humane manner and that the work is worth it. Shorter lifetime, yes, indeed. Worms have a shorter lifetime than humans. They live about 17 days, I think, whereas we live up to 70 to 90 years. You can do more controlled studies in model organisms than you can in humans. Yes, we can genetically manipulate them and do really refined experiments that we simply, even if it was ethical, which it's not, even if it was ethical, it wouldn't be possible to do in humans. Easy, start to know, we know a lot about each model organism, faster generations to study, shorter generation time. You're on it. This is great, thank you. So here's the worm. I'm now going to take you through what the features of this particular model organism are, and you'll see some of the features that you just listed as why we do our studies in model organisms. So this is your pronunciation guide, Caenorhabditis elegans. The worm is small, as I mentioned, 1 millimeter long. The worm is simple, but it has all of the major tissue types. Muscle, nerve, intestine, skin. There are only 959 somatic cells. And by that, I mean non-germ cells, non-reproductive cells, like muscle, nerve, intestine. What's so special about 959 is it's a small number. And we know the number. We know exactly 959. 32 cells in the gut, 302 cells in the nervous system, 85 muscle cells. We also know the lineage. In other words, all cell divisions from the embryo to adulthood, or certainly to larval development, have been tracked. And so we know what every cell develops into and the fate, what every cell develops into and where every cell came from in the adult. And that's incredibly powerful information to have for developmental biologists. Worms are easy to culture. In the lab, we grow them on bacteria. In the wild, they grow on rotting fruit. As we've mentioned, they have fast development, a 3 to 5 day life cycle. So from the time they're a baby until they grow up to make babies. They have excellent genetics. We can make mutants and analyze defects. And for those of you who think about or know about genetics, we can do both forward and reverse genetics in worms. We can come back to that in questions if you want to. And they make lots of offspring, about 300 offspring per worm. When I went to talk to middle schoolers about genetics and epigenetics, the feature of worms that they found most remarkable and interesting was that you can freeze worms. We can freeze them down, store them for years, and then thaw them and keep working on them. Amazing. For the purposes of our studies, it's important that across many species, there is conservations of genes and developmental pathways across worms, fruit flies, humans, and some of those genes and pathways are better conserved than others. But the hope is that experiments that we do in model systems will inform experiments in higher organisms like humans. So to give you a preview of what this talk will be about, I'm going to be talking about passing what I call an epigenetic memory of germline from parents to offspring. So here we have a mom worm, her germline is in purple, a dad worm, his germline is in purple. Genes, so our model is that genes that are expressed in germ cells are marked with an on mark that gets passed to offspring. And genes that are kept silent in germ cells are marked with an off mark, and that gets passed to offspring. And that the passage of those marks through the egg and the sperm into the embryo and through cell divisions to these cells that are the founding cells for the adult germline, they are the primordial germ cells. We think that those on and off mark memories help tell those cells how to develop so that they develop into, so that the germline develops into a fully functional, fertile animal. Now, I said before that the typical epigenetic mediators that many labs study are DNA methylation, histone modifications, and small RNAs. Worms don't do DNA methylation, so that one is off the table. And the one I'll focus on is histone modifications. Worms certainly use small RNAs, and we're trying to understand the interplay between regulation by histone modifications and small RNAs, but I'll focus today on histone modifications. Okay, so we're back here, epigenetic information, and now I want to lay out the whole concept of gene expression and on and off marks. So I'm creating two models. One is gene X that has an on mark, and the other is gene Y that has an off mark. And I mean that on and off mark are scattered along the gene. What I mean by gene X having the on mark and therefore being expressed is that gene X is transcribed into RNA, which is translated into protein, and then the protein carries out the function of the gene. For some genes, the RNA is what carries out the function, but for many genes, it's the protein product that carries out the function. So if gene X is on, what we mean is that it is made into RNA and likely protein. If gene Y over here is off, what we mean is it is not transcribed into RNA and it is not translated into protein. Now, be aware that all cells in your body have both gene X and gene Y. So it's a matter of how those genes are transcribed in different cells that make those cells different. For instance, in muscle cells, genes that are on are those that make actin and myosin and allow you to contract your muscles, right? In germ cells, the genes that are on are genes that are necessary for the specialized cell cycle of meiosis and for making eggs and sperm. And in germ cells, they want to keep muscle genes off, okay? So all cells have the same set of genes, but how they deploy them, which ones they transcribe and translate is what dictates tissue development. Do the on and the off marks matter? And the answer is a whopping, resounding yes. And this comes from our genetic analysis. A normal mother that can provide on and off marks to her offspring enable those two primordial germ cells. They inherited the memory to develop into a full functional germline. But a mutant mother, and we have these mutant mothers that cannot make the marks. So they cannot put marks on the genes that need to be marked as on and off. They cannot pass that memory into the offspring. And as a result, these two primordial germ cells die a few divisions after they're supposed to launch their program. And the resulting adults are actually big, happy, healthy adults, but they have no germ cell tissue, no germ cells. Now, one feature of worms that's really special is we can see with our eyes under a microscope if worms are fertile, they have embryos in them, or if worms are sterile, they don't have embryos in them. And that greatly facilitated our isolating the genes that put down these marks. In most other organisms, you have to do matings to tell if an animal is fertile or sterile. So I want to give you an overview of what some of my lab's major advances are, and then take you into how we actually did one particular study. So through our genetic analysis, we identified the epigenetic on and off marks. And for those who know about histone modifications, the on mark is methylation of H3 K36 on histone H3. And the off mark is methylation of K27 on the tail of histone H3. And those are yin-yang marks. So a histone tail will have an on mark or an off mark. They're mutually exclusive. Through our genetics, we identified the writers. In other words, the enzymes or proteins that write the marks. They're the ones that put the marks down on the histone tails. And we tracked the passage of the on and off marks from parents to offspring, and we tracked them on through cell divisions to the primordial germ cells. And it's these latter experiments that I want to tell you about. How did we do this? How am I doing on time? Good. So how to see the off marks. Here are on marks along this region, and I've just color-coded them yellow. And here are off marks along here, and I've color-coded them blue. And I'm asking how to see the off marks. And we're going to focus on the off marks. H3, K27, methyl 3, for those who know the marks. The way we see them is to use antibodies. So these are little Y-shaped molecules that are in your immune system and that help your body recognize whether things that you encounter in the environment or in your food are self or foreign. Are they foreign and should be fought off, or do they belong in your body? And so we can obtain antibodies to the off marks. We actually buy them from a company. We didn't even have to make them ourselves. Now, antibodies are not visible. So we have antibodies, but we haven't yet made them. We haven't been able to see them yet. And so the way we make them visible is to tag them with a green molecule. So imagine a green glow stick, right? Bright, brilliant green glow stick. That is attached to those antibody Y-shaped molecules. And that causes the off marks along the chromosome to glow green. So here I'm showing green all along this region that has the off mark. And there are other regions in the nucleus that have the off marks. Now, if we were doing this experiment, and I spent lots and lots of time doing experiments like this, it's called immunofluorescence microscopy. I would be in a dark room and I would look for the green marks, and it would look like this. So you're saying, well, how do you know where they are? And it's because we actually have all the DNA marked red. So I didn't mark that here, but basically what I'm looking for is green along a big swatch of red DNA. All right? So we're using green marker, and this is actually the green protein that we use is green fluorescent protein, GFP from jellyfish. We use that a lot, in part because when we want to do those experiments with GFP, we can do them in live animals, whereas sometimes we use other colored markers when we want to be working in fixed animals. So either way, we're working with green glowing antibodies. So what does the pattern look like on chromosomes? And this is a micrograph that we generated in the lab, and it shows 6 chromosomes here that have red stained DNA and lack the off marks. They don't have any green. And we have 6 chromosomes here that have red stained DNA, and they have the off marks. And you can see that when you mix green and red, you sort of get yellow. That's the mixture that you get. So with this sort of strategy, we did this experiment. We asked the question, do sperm, we took them a germ cell at a time, do sperm pass off marks to the embryo? So we did sort of strategic matings where the father was normal and could put off marks on his chromosomes. And I know this doesn't look like a sperm to you, but worm sperm are very unusual. Instead of having a flagellum, like many sperm that we're familiar with do, they have a pseudopod, and they sort of crawl along the surface. Sorry, you can't see my fingers. They crawl along the surface. And then we had a mother worm that was mutant and could not put the off marks on her chromosomes. So her chromosomes lack the off marks. And then we said, if the off marks are passed on chromosomes to the embryo, we should see a pattern like this in the one-cell embryo. Sperm chromosomes that have the off marks, egg chromosomes that lack the off marks. And that is what we saw. And this is the image I just showed you. These are the egg chromosomes that lack the off marks and lack green staining. These are the sperm chromosomes that have the off marks and have green staining. And you might be wondering from my last cartoon image, why does the green look so uniform along the chromosomes? Why doesn't it look more spotty? And we know from higher resolution analysis that it is spotty, but this is low resolution imaging that is just giving us a snapshot of chromosomes overall. To crawl in and really see the gene level distribution, we do a type of approach called CHIP, for chromatin immunoprecipitation. And that allows us to see the on and the off marks at gene level resolution. So we see on marks, off marks, on marks, all the way down the chromosome. Now, if we do the opposite experiment, this is what I just showed you. Now we do the opposite experiment over here. And the difference is now the mother is normal, she's wild type, and she puts off marks on her chromosomes. The father is mutant. He cannot put off marks on his chromosomes. And now we expect the marking in the one-cell embryo to look like this, present on the egg chromosomes, absent from the sperm chromosomes. And that's what we see. Egg chromosomes with the mark, sperm chromosomes without the mark. So this is the first step of memory. You need, for these off and on marks to be providing memory, you need to pass the marked chromosomes from the parent germline into the offspring. And we just accomplished that, into the one-cell embryo. And now comes a big challenge. How, I guess, are the off marks transmitted through DNA replication? And by DNA replication, I mean copying of one chromosome into two daughter chromosomes. And I'm going to show you a cartoon slide to try to explain this. Although of all the parts of my talk, I think this is probably the most complex. So if this is hard to follow, then I think you can get the rest of it, but I want to try to put the mechanistic underpinnings here. So I'm showing you a chromosome here that has marks, and I've just made them into red flags. This could be any mark. This is one chromosome, DNA wrapped around those beads, nucleosomes. And this is the machinery that's going to march leftward and make a copy of this chromosome into two daughter chromosomes. So this chromosome will basically be taken apart and made into two. And what happens, the model was that these beads, these nucleosomes, get taken apart and they get passed back to the daughter chromosomes. So that the marks that were here to begin with are in that same region on the daughter chromosomes. So this would be the model for how marked chromosomes create marked daughter chromosomes. The other part of this is that when chromosomes start off unmarked, they produce unmarked daughter chromosomes. That would be showing faithful transmission of these histone marks. So let me show you the test we did. As embryo cells divide, are the off marks passed to daughter cells? That was the question I posed. We got to this point before: 6 sperm chromosomes with green marking, 6 egg chromosomes without green marking. Each one of these chromosomes is now copied into 2 daughter chromosomes that are stuck together. But all of these chromosomes will intermingle in the middle of that one-cell embryo. And then when the one-cell embryo divides into 2 cells, each cell gets an equivalent portion of those chromosomes. So if the daughter chromosomes from these inherited the marks, we expect those daughter chromosomes here to show marking. If these chromosomes lacked the marks and didn't acquire any new marks, we expect them to lack marking. And that is what we see. So this is showing you one nucleus here that has 12 chromosomes. 6 were derived ultimately from these. 6 were derived ultimately from these. And you can see that the 6 that were derived from the sperm chromosomes retain the marking. So the chromosomes that came in marked generate daughter chromosomes that maintain marking. And this pattern persists through many cell divisions, which I haven't shown you here. The nuclei get smaller and smaller, but they still remain very much red and green. And this marking is sent all the way to those primordial germ cells in a manner that we think shows that memory has been passed. So, as I said, the off marks stay confined in this case to the sperm-derived chromosomes. That was our test case. If we did it the other way, they would stay confined to the oocyte-derived chromosomes. And this demonstrates memory. It demonstrates that memory was inherited, often on marks, in this case, off marks were inherited from parents, passed into the one-cell embryo, and then passed through DNA replication and cell division to maintain that memory. So the significance of this is these findings in worms provide visual and really compelling evidence that important epigenetic information can be transmitted from parents to offspring. I'm in a field where many of the studies are biochemistry, really elegant biochemistry. And the first time I presented this story was actually at a meeting in Heidelberg, Germany. And the other speakers and the audience were not used to looking at genetic and microscopy studies to tackle this question. And it was a profoundly rewarding experience to have everyone be so excited by being able to see this. We also showed that the information after being passed into the one-cell embryo, that it's passed via both egg and sperm up here, and it's transmitted through cell divisions, like I showed you in the second set of slides. Now, we did an experiment here that I'll just summarize. I won't show you the data here. We know that if we take away all the off marks from both sets of egg and sperm chromosomes, that these two primordial germ cells cannot get off the starting block. They're really impaired. And so we wanted to do a more refined treatment. And we thought, what would happen if you just remove the off marks from the sperm? So send in off marks on the egg chromosomes, but don't send in off marks on the sperm chromosomes. What happens to development of the offspring? And what happens is when, in the absence of information in sperm, they're lacking the off marks. That causes gene expression in the germline of the offspring to be impaired. And remarkably, it causes germ cells to reprogram toward neurons. In other words, these animals, they have what might be considered a germline, but they're lacking functional germ cells. And they have cells instead that send out processes that look like neurons and that have neuronal markers. That, to me, is surprising. I think of neurons as being such specialized cells. It wasn't clear, and still maybe isn't clear, why germ cells would reprogram toward those. And I think the answer must lie in the fact that when sperm chromosomes come in unprotected, in other words, they do not have, say, neuronal genes covered with off marks. And in that case, those neuronal genes can fire. They can turn on in these cells and overwhelm the system and cause the germ line to lose its germ cell identity and instead go toward neuronal fate. The implications for humans, indeed, the on and the off marks are used in diverse animals, including humans. So our on mark, this lysine 36 methylation on the tail of H3, the off mark lysine 27 methylation on the tail of H3 are widely conserved across species. And so are the writers that make those marks. So when we carried out genetic screens to identify these, we identified genes that were known in other systems to do what the other systems use those genes for. Loss or overproduction of those writers in humans causes all kinds of developmental defects and contributes to many types of cancer, including pediatric gliomas, lymphomas, and adenocarcinomas. So what I've tried to convey is that Overcalyx and other studies have shown us that the nutrition of parents, their health, whether they smoke, for instance, and the nutrition and health of parents and even grandparents can influence the health of their offspring and grand offspring. And it manifests as a different an effect on the incidence of heart disease, obesity, and diabetes. We really hope that our studies in worms they're not a precise model for certain things that happen in humans, but they are a model for how epigenetic marking and memories can be transmitted from parent to offspring. And our newest generation of experiments are suggesting that the marking and consequences can even be passed to the grand offspring in worms. So we're sort of trying to traverse the generations in worms. Good. So I would like to thank my favorite teams. Yeah, we have plenty of time for questions. First and foremost is my lab. I have a great group of researchers. This is when we were all gathering at Sabing to eat Thai food. This is the current Strome lab, and I have quite a few recent alums from UC Santa Cruz who contributed a lot to our ongoing studies. Laura Gatos is the graduate student who did the do sperm transmit the off marks analysis. And it was really well received and published in a nice paper. I want to thank my colleagues in MCD biology, molecular cell and developmental biology. I have a great department and we all talk and interact a lot, except this guy, he doesn't look so happy. Everybody else seems pretty pleased. And then I would like to thank the home team. So this is the young, me and my husband and the young kids. And this is a more current version of us. I am a woman in science. I'm a WIS, and I've given various talks at women in science conferences. Importantly, I have a husband in science. And now guess where I'm going? I have a daughter in science. Sarah is a graduate student in bioengineering at the University of Washington in Seattle. And I have a son in science. He is a graduate student at Berkeley studying epigenetics in yeast. So he loves to come home on the weekends, surf with his dad, talk epigenetics with his mom. And then what would you name a dog that belonged in this family? You would name it Lab. Okay, I will stop there and I'll let Colin start feeding me questions. Maybe I can see these. I'm going to do Command, Tab. There, try this. Okay, I am going to start reading some of the questions that I can see in the presenter chat. Okay, are you seeing them here? Or are you still seeing my screen?
We're still seeing your screen.
Okay. I'll leave it here, although if it seems like we don't need to have the screen share anymore, I'll turn it off. Are there other histone modifications beside these on and off marks? And if so, what are they? Is there a histone code? Yes, there are a myriad of other histone modifications. There are other residues on the tails of mainly histone H3 and histone H4 that get methylated. And there are residues that are acetylated and phosphorylated. And there is a histone code. Combinations of marks can reinforce each other and have impacts on gene expression. How many generations could a change on-off be transmitted? And the question is indefinitely. So I don't think it would last more than a few generations, the type we're following. Histone modifications, we have evidence now, some evidence that it's going into the grand offspring. I don't expect it would go much longer, and if it does, I think it would start becoming diluted. There is some evidence from another lab in Spain for some persistence beyond just that first generation of offspring. Now, RNAs, as an epigenetic mediator, I think have much more potential to be passed over many generations. One analysis suggested that RNA effects could last up to 17 generations. The effects might become gradually diminished, but there's definitely persistence. Does this imply that older parents with more health problems are more likely to pass on health problems to kids? Let me take that apart. Parents with health problems may pass on a leaning in their children toward having similar health problems. I am diabetic. My parents are not diabetic, but my sister is. I'm pretty sure there's a genetic component to that. There may be an epigenetic component to that, but now it's pretty well understood that that disease is a myriad of genetic markers that make me predisposed, and then there's a trigger, and then my autoimmune system kicks in and kills my pancreas cells. Many health issues, I think, have a genetic underpinning, and there can be passage of some of that to offspring. Do older parents? Yeah, older parents. So older parents start to have more incidences of miscarriage and issues because they're, especially mothers, but also fathers, because their oocytes were made early in development and so have been in the ovary for decades, and they start deteriorating. And that can lead to oocytes that don't work and are not actually fertilizable, oocytes that may be fertilized but may not develop normally, and then oocytes that are fertilized and develop, and the individual, I started to say hatches out, but the individual, the human, is born and might have health issues. So an example of that is Down syndrome, which is due to chromosomes suffering more and more deleterious effects as mothers age. Oh, I see what you're doing. Let me see, I'm trying to get to. Have religious groups with different mandated lifestyles been studied for differences in epigenetic differences? I don't know. That might be a rich resource if you have a whole population that has a lifestyle that would allow you to compare, that you could compare to another lifestyle. It's more typical to try to do that kind of experiment in a model system. For example, in mice, you can sort of mimic what you might be asking here. You can alter diet, you can alter what mice, what stresses and toxins they're exposed to, and then directly analyze the consequences. And Upasana Sharma in my department, you should ask her for a Café Scientifique talk. She has a wonderful story that when father mice are fed a low protein diet, that it impacts the small RNA population in their sperm. And then the sperm fertilize an egg, and the egg didn't come from a low protein mom. It's just the father that had a low protein diet. And there are consequences in the embryo. So this is a small RNA story. This is not histone marks, although maybe it involves histone marks. But it's mainly small RNAs, and it's actually tRNA fragments, for those of you who might think about small RNAs. So one of the really interesting aspects of her study is how does paternal diet alter the RNA payload in sperm? And she has evidence that it alters the RNA in the somatic tissues that surround the sperm. So somatic tissues, remember, are the non-germ cell tissues. So these are not reproductive, but they surround the reproductive cells that give rise to sperm. And that the small RNAs are sent from the somatic cells, it's the epididymis, in little vesicles to the sperm, and then they download their RNAs into the sperm. And that might be the mechanism by which a dad fed a low protein diet can impact what small RNAs are in sperm, and in that way impact gene expression and development in offspring. It's a complex but fascinating story. How would environmental factors like diet influence on-off? So ultimately, influencing on-off, you can influence it in the generation that's being studied, or it can be influenced in the germ cells and pass that influence to the offspring. Now, there's one, this isn't diet, but there's one interesting study that's been done in worms that's about salt. If you subject worms to high salt, if it's really high salt, they go into a rest until they can adapt to it, and then they come out of it. But if you expose them to intermediate salt levels, then they can send a message from the neurons that sense the intermediate salt level to the germ cells and cause the offspring to be more resistant to high salt. So it's a really nice example of epigenetic transmission of an environmental influence experienced in the parent that impacts how the offspring respond to that same influence. A lot of the steps of that are not known, but some of the steps are. Can one worm tell if another worm is sterile or fertile, or whether to try to mate with them? Or are they indiscriminate in their mating? That's a good question. Let's see. A worm, when we're mating, males may know more than I think they know, but when we mate males, so let me tell you something first. Worms come in two sexes, males and hermaphrodites. So hermaphrodites make both eggs and sperm. Males make only sperm. When hermaphrodites don't have any males around, they make eggs. First they make sperm, then they make eggs, then they fertilize their eggs with their own sperm. They make embryos, and then they lay the embryos out on the auger. So embryos can be inside the worm, and if you now bring in a male and say, go mate with these worms, the males are capable of mating with hermaphrodites that have embryos inside them, hermaphrodites that don't yet have embryos inside them, or hermaphrodites that we've rendered sterile. I think they still try to mate with those. So I'm not aware that they're discriminating. They may, though. There may be subtle differences. Can ailments such as depression or anxiety be passed on? I would say sometimes there's a genetic component to that. And often the genetic component is not as simple as a gene. It's more that a certain combination of gene alleles leads to a predisposition. So I would say genetic underpinnings are there. Epigenetic, maybe. The lab that Upasana Sharma came from has done experiments where they make mice anxious. So they're mimicking sort of depression or anxiety and asking whether that is passed on to offspring. And it is in some form. So yes, there is some evidence for an epigenetic component too. All right. Are those my questions? Have you seen a strong correlation between methylation of histone and the corresponding region of DNA's exposure to transcription that passes down the generations? Well, there is a strong correlation between the methylation pattern of a DNA region and its transcriptional capacity, for sure. Now, sometimes we're a little bit confused about what's cause effect there. In other words, is it that having on marks leads to that region being transcribed? Or does that region being transcribed lead to that region acquiring on marks? But definitely there's that correlation. And when I talk about our studies now following into the offspring and grand offspring, I think we're seeing that correlation be passed on for at least those 2 generations. is evolution an accumulation of epigenetic on-off switching of genes? There could be some epigenetic component, but I think about evolution as being, as having a stronger genetic underpinning. So mutations occur, and if those mutations confer an advantage, then they become more prevalent in the population. And over time, organisms evolve, and you can even get speciation, where they develop into diverse species and cannot cross-mate. One of the features of epigenetics is that the marks are reversible and flexible, and that's a particularly beneficial feature. Epigenetic marks would allow an organism to respond to the environment right then and there, whereas evolution is over generations, long periods of time. And then the question becomes, would epigenetic marking, for instance, be permanent? I don't know. I do consider that to be more a genetic process. I'll go talk to my epigenetic colleagues about that. Oh, do you think your sons and daughters' science proclivities, nurture or nature? Could math science talent be genetically influenced? Maybe there's a genetic component, but there's certainly a nurture component. I mean, when they were in science classes in middle school and high school, we would lay out on the kitchen table bananas to be endoplasmic reticulum, with raisins on top to be ribosomes. We were doing all of this extra tutoring and guidance that I think made it comfortable for them to think about science. So that's nurture, and maybe they inherited a love of the type of science we do as well. One thing I do think they saw, and this isn't necessarily related to your question, is they witnessed that they had two scientist parents who we love what we do. And that's a really powerful message to send to kids. And furthermore, they could see that what we do is we're lifelong learners. You know, it's not like we stop learning once we start actually teaching. In fact, that's when we go into hyper learning mode. So I think that was a powerful message for nudging them further towards science, is seeing us, my husband and me, enjoy our science careers. Last question. Could epigenetic changes contribute to the acquisition of antibiotic resistance by bacteria? I was going to say, well, so bacteria do not have nucleosomes per se. So the kind of epigenetic marking that I'm talking about, I'm not aware is in bacteria, unless there have been new developments that I haven't kept up with. I don't know the answer to this. My tendency is to say, sure, epigenetics can be involved in a lot of interesting phenomenology. Antibiotic resistance. I mean, most antibiotic resistance is bacteria figuring out how to evade the point inside the cell where the antibiotic functions. And so it's often a matter of, if the antibiotic affects the ribosome, then a mutation in the ribosome confers resistance, and then they're resistant. Or if an antibiotic affects influx of things through the membrane, then a mutation can confer resistance. So I do tend to think of it as being more genetic based, but there may be an epigenetic component. Leave the door open. All right, I thank you for hosting me, and this was fun, talking to my screen and imagining the faces out there.
All right. Yeah. Thank you, Susan. I think we'll wrap up here. And thank you, everyone, for tuning into our virtual Cafe Sci this evening. I think we all learned something new about epigenetic inheritance tonight. So I'm glad you were able to join us tonight, Susan. Thank you. For the audience, I hope to see you all back in a month for our next virtual Cafe Sci. So be sure to register through hanahouse.com or on the Cafe Sci website. Have a great rest of your Thursday evening, and thank you again for tuning in. See you guys next time.